Yeah, so tonight, Gregor, who is a theoretician of visual languages, will be presenting his project DNA Semantics, which he started work on already a few years ago when he was a scholar at MIT in 1984. And yeah, since then, he showed it, he started showing it 15 years ago in the form of an exhibition titled Genesis, which, as Gregor was saying, was shown in galleries such as Henry Art Gallery and such as, I have it here somewhere.
It was Berkeley University, and there was another third place, I forgot. To tell you the truth, it was a long time ago.
Yeah, block.
But it was shown. You find it in the information, so it is there.
Yeah, let me see. Yeah, at Berkeley Art Museum in California and at Weizmann Art Museum in Minneapolis, and Mary and Lee Block Museum of Art in Illinois. So it's presented for the first time in a comprehensive way in the form of the book DNA Semantics, which was published last year by the Visual Language Research Institute and available tonight for sale in the back, so make sure to get your copy. And also in the form of an exhibition of the same name, DNA Semantics, which is currently on view in Beyond the Globe, the 8th Triennial of Contemporary Art in Ljubljana, which is curated by Boris Groys. And so thank you for coming, and let's give Gregor a warm welcome.
Thank you, Anna. So we'll start with this very familiar representation of DNA as a double helix. We have this it has in essence two parts. One is this backbone that is called, that is sugar phosphate, and there are these bonds between that are called the base pairs. There are two base pairs, four bases. And the base pairs are between bases adenine and thymine and guanine and cytosine. These are the base pairs. So this is kind of a fundamental structure of the DNA. Yeah. Okay, I did that. However, DNA is a part of, in fact, two structures, DNA and RNA. And RNA, as you can see on the right, is a single strand. It's not a double helix. But they share, both DNA and RNA share three bases. Adenine, cytosine, and guanine are in both DNA and RNA. Only uracil is in RNA, and thymine is DNA. That makes distinction between these two kind of strands. Now, these are chemical structures of these five bases. Four that plays a role in the DNA and four that plays a role in RNA. And we see that there is one kind of a ring hexagonal structure pyramidine in which are uracil, thymine, and cytosine, and another is double ring structure purine, and in which are adenine and guanine. Thanks to their properties, adenine and thymine, since they form the base pair, the base pair is formed with two hydrogen bonds, and which we can see here. While guanine and cytosine are establish their pairs, like a bonding, pair bonding, as through three hydrogen bonds. So that's what makes the difference between these two base pairs. So, one important property of the DNA is replication. DNA can replicate. It can either you can think as one or original DNA making a copy of itself, identical copy, or you have other way to tell it. It would be that we got from one DNA double helix, we have two DNA identical. As you can see here, T and A are identical here with T and A. These are identical twins in essence. That is called replication. Another kind of a feature or phenomenon is called transcription. And transcription now involves another strand which is called RNA. And RNA is a single strand. What does it mean? It means that at some section, DNA, so it's here, like DNA would open and one part would be copied by RNA. So we get a picture of it. So this one is non-template strand, and this one is called a template strand. And in essence, what is copied is a template strand. But in fact, we can think that we can assume that the real code is here, and it is pictured here and then copied on RNA. The reason why we can see here in the next picture, which is a better illustration, we can see that this is one side that is a template DNA, and we see RNA copying it. And as you can see, C is copied into G. These are base pairs. G is copied into C. T is copied into A, and A is copying into uracil. However, if we compare this RNA with this non-template or non-coding DNA side, we see that it is almost identical. We see G is G, C is C. Only uracil and thymine are different. That's the only difference. So if their entire operation is made in order to turn thymine into uracil, that's all. Everything else remains the same as here. And that is I'm pointing this out because in the method that I'm proposing here, there is an explanation for this. So the first image of DNA was, recorded by Maurice Wilkins and Rosalind Franklin, 1953. And this is through the X-ray crystallography, and it indicated, like this X marks in X shape, indicated that it is not just a simple strand, it is a double helix. And James Watson and Francis Crick came up with this double helix structure. This is the earliest version, which today becomes multiplied in various combinations and variations, as we can see here. But these are all models. This is not DNA. This is just a more interpretation. Even we can think of the X-ray picture as an interpretation, but more direct. So this for many decades, this was the only picture of DNA. Only in the recent years, with the introduction of electronic microscope, it was possible to get a close-ups of DNA. And what we see here are some 40 nanometers close-ups of the sections that shows DNA. This is a model imposed on the part that, in fact, is beneath is a DNA, a picture, or something like this, or this. This is the most recent picture of DNA, actually. And we can see here, marked with this red arrows, the helix, double helix. So it was done by Italian team, and this is the closest image of DNA we have today as a picture. So on the left is a picture of DNA, on the right is the model. And this is another model. Here we have introduction of the colors, as you can see on the here. But as you would notice that all the bases are represented with the same color, dark blue. Here there are bright blue, but again the same color. Only the shapes, they make difference. As you can see, the dots are showing the double hydrogen and triple hydrogen bonding. And now we have cases where each of the bases is presented with a different color. We have a key here. And I will show you now many of these. These are just a small selection. And what you can get from these color coding is that it is in essence arbitrary. There is no standard in representing bases today in any considering the color coding. For each one is different. For example, here adenine is orange, thymine is blue, and here adenine is green and thymine is again blue. And here adenine is red and thymine is blue. And now we have large, since the strands are really long, they are long, very long. It is a problem how to present them, that we can handle them. And one way to do it, to treat them the way we treat the sentences in the book. We just decide what's the width of the book, and then we make this as a two-dimensional images. So these are all kind of arbitrary two-dimensional representations of long DNA strands, which in fact are not based on any kind of a rational foundation. It is in essence this. A long could be, you will see later I did a E. coli strand that was 4,600,060 positions. It's a long line. So, but it has to be presented this way. But what we see is, as a two-dimensional is completely arbitrary. So the question was, is there any way we can represent the bases that are not only turned into this linear structure into two-dimensional, but also to use a different way to represent these bases, not to use these initials of the names of the bases, as T is for thymine, adenine, A is for adenine, and so forth. This is completely arbitrary. And this is the proposal. Here we see what we have today, alphabet representation of genetic code in RNA form and one of the DNA forms, and the visual representation that I would be presenting it here. And clearly, if you look to the left and to the right, you won't see on the first site, you won't see much difference. But when you look these two images, you see a difference. They tell you more than what the letter representation is telling you. They show some structure and they show some patterns. That is what makes the difference. So you can, and this is the proposal. The proposal is to, instead of using these 5 arbitrary letters, we should, I'll say, assign these 5 discrete values of the grayscale to each of the bases by this proposition. There are 120 ways to connect 5 letters with the 5 values, but only this one and its negative, and we'll go to the negative issue, will show we are actually the most accurately representing DNA. Others are not good. So in this representation, uracil is defined as 100% black. Black is a value that we don't define. We can say it's out of the system. We can say it's absence of light. That's black. And this is 100% black. 75% black is cytosine. Adenine is 50% black, which means gray. Guanine is 25% and thymine is 0. So the neighboring values are 25%. Now, if I would ask you to find the pairs that differ 50%, would you be able to identify them? Which bases differ 50%? A? Yeah, A and, but A, which pairs? Yeah, exactly. And these are all base pairs that exist. There are no others, there are no less and more. With this single rule, we have established all three base pairs, 2 for DNA and 2 for RNA. In order to do it with letters, we have to list all three. Here we just have one rule. So this shows that there is some underlying property that these values represent here. And so these are, so we have T and A and U, and G and C. But in today's representation of base pairs and understanding, interpretation of the base pairs, the base pair is a relationship, like bonding. But with this representation here, we have additional element, which is that it's not just bonding, it's a transaction. It's an exchange. And exchange is that if G and C are base pair, their transaction from G to C is addition of 50%, which means addition of value of adenine, symbolic value of adenine. If we have CG as a base pair, it is subtraction. So we have to subtract value of adenine in order to get guanine. And same thing here for thymine, which is a 0% plus 50%, we get adenine. And that's T and A are base pair. And A and T again, from A to T, it is a subtraction. So instead of having just simple bonding, we have transaction. And what's interesting, that is the adenine, which value is 50%. It's a gray. That is that currency that is being exchanged. That's interesting. Now, this is, as you can see, the difference between the bases, as we said, between this representation of the bases is 25%. And we can even think of, for another purpose, but not for this one, that we can treat this relationship between, let's say, cytosine and adenine as some kind of a transaction of 25%. But we have to find a phenomenon for this. It is not, in essence, so far, could not be identified with the DNA. We will now introduce a concept of negative. Negative is something that is very visual. If you know from the even time of photography, analog photography, you have a positive of the film, photography, and you have a negative. So everything what was black in the negative becomes white on the positive, and vice versa. That's photography. here we see that we will define the negative the way that a negative of uracil is a white thymine. Negative of cytosine is, which is dark gray, is light gray. Negative of adenine is adenine. Adenine is, or a gray, is neutral to the operation negative. It doesn't change. So, it's a very interesting property. And adenine is the currency that is being exchanged as well. So, this is the, how to say, definition of negative. This is for each individual base. So, we have thymine into uracil. It's 100% difference. And from uracil to thymine is 100%. These are not base pairs. These are just transactions or operation that was called negative. Then we have a light gray and dark gray. Light gray, negative light gray is dark gray. But this is also a base pair. Guanine and cytosine are the base pair. So here the base pair relationship and positive-negative relationship completely coincide. And that will have some role to play. And finally, we have a gray is equal to gray. Gray doesn't care about negative. Now, when we observe now the DNA and RNA as a subsets of this 5-element gray scale, we will notice that they relate to each other as positive and negative. DNA and RNA are like positive to negative. And these are all base pairs. Like this is a base pair, I mean not all. This is a base pair and this is a base pair. This is gray-gray, copied gray into gray, and this is white into white. There is, unlike in the case of base pairs, when you had only 50%, a single one exchange value, here we have, for black and white, we have 100% difference. And for gray, we have a 0% difference. So it's not constant. Now, if this is like a non-template, non-coding DNA part, and this is a coding DNA part, and this is RNA that is in fact taking the code, copying the code from the coding part, what we have here is that guanine would establish a base pair with a cytosine with a 50%, and then go back to guanine. And these are all 50% transactions. But to get from negative of thymine, we cannot do it in one step. We cannot, because we have a limitation of 50%. So in order to get negative of thymine, we have to jump twice. And this is why we need both replication and transcription. Because from here to here would be replication, and from here to here would be transcription. So this explains that why we need two kinds of transactions. In order to get negative of thymine, to get uracil, black. But it cannot be done in one step, it has to be done in two steps, because there is a 50% limit. So that is something you won't see with the alphabet representation. It doesn't exist. So in a way, now the bases that we see them as simple letters have in fact some kind of a hierarchy. Here, thymine and uracil are in fact the key players. And adenine is in a way go between. It's something that plays like a mediator between uracil and adenine. And G and C are in some kind of a supporting role. They just, I would say, add complexity. If I would have to anticipate the most likely these three were the oldest bases. The T-min, in fact, it was probably binary at some point, and then became trinary, and the G and C just were added to how to say, make more complexity, to add to complexity. So, at this point, we have 4 kinds of representations of DNA. One, which we are all familiar, which is this letters representation, which doesn't tell you anything. And then we have this grayscale representation that you can see the relationships in values between the bases. Then you can instead of having letters, or the grayscale, you can use percentage values. So this is equivalent, whether we use a 0 or T, it doesn't matter for the structure. But the 0 tells you something that T doesn't tell you. Or we can use the simple numbers 0, 1, 2, and 3 to represent all 4 bases. And you will remain all these relationships that we have for the visual representation. But the visual representation is much more, attractive, because it gives you an image. And for example, it has this property of negative. You can say that negative of dark gray is light gray, or you cannot say with the numbers. But numbers are important in some cases when you need a quantifying element, when you want to calculate the value of a certain sector. You can count it. You cannot count it with letters. But you can say, for example, if we add all the numbers that are on this bottom line, we can have the quantifying value of this stretch. 2 plus 0 plus 1 plus 2 plus 1 plus 3, and we'll have a number. Whether it will be of some use or not, we don't know. But it's something, it's another additional information. So that is what we have with this visual representation. Until now, I was mostly telling you about values. So these are this is a structure of values. We have 5 values: black, dark gray, gray, light gray, and white. Values have no special relationships. So here, let's say, dark gray and gray are neighbors in this structure. But dark gray can be in this corner and light gray can be on that corner. They are still neighbors. Their neighborhood relationship absolutely is irrelevant to where they are, because they are structures of values. They are here placed into certain structural positions. That's why they look like, it looks natural, but they are, in fact, it's absolutely unnecessary, because, and this is from this work I mentioned you in 1984, discrete visual structures, when I managed to separate the structural position and structural values. In structural position, when you see on the left, we don't care what is the content. We just care how they relate to each other spatially. When I'm using a space, I'm using a 2-dimensional space here. It's not 3D. And here we have a structure of values. It's irrelevant how they are positioned to each other spatially. But when we bring them together and when we put these values in these positions, then we get a complex image, what we have here. By the way, this paper you can find, it's on the MIT site under the name Discrete Visual Structures, Element of Visual Grammar. So, and to apply this distinction between structures of value and structures of position, we can show how this, what I've been showing so far, this code, how to translate a letter representation in visual representation, is in fact consists of 3 parts. We have a structure of position, 5 elements, linear. We have a structure of values, and as you can see, they are not neighboring. So light gray is neighbor to white, but it doesn't have to be next to white, because they are neighbors by values, not by position. And then we have also another set of 5 letters that are attached to this. So this picture, in fact, here on the left is consisting of 3 structures that are brought together. It's not a simple one. So once we have established now the structure of values and their relationships to the bases, the 5 bases, the question is there any way to translate in a meaningful way the linear structure into 2D image that is also meaningful? And there is a linear structure already existing the spatial structure that exists in the DNA, which is called codon. Codon is 3-element linear structure that is has one neighbor on the left, one neighbor on the right, and middle has 2 neighbors, both left and right. Codon codes for the protein. And there are 64 codons in the genetic code, out of which there are 24 proteins, and some proteins, as you can see here, they appear in 4 redundancies. So we have here kind of a 3x4 structures. That's entire genetic code. And these are some of these proteins that are appearing so this is the same. These are 4 codons will produce the same protein. So as I said, we have in essence this one line which is consisting of a series of codons, and we want to translate it from this into this. So already we've seen this 3x4 structures that appears in the way how certain proteins appear in genetic code. We have now 4 codons placed one above another as a 2-dimensional structure. And the point here is that we will have, let's say, if we start here, we will place 3 codons, then we will start here, we'll place another 3 codons beneath, another 3 codons beneath, and another 3, and we'll have 3x4 image. Then we will start again a new one. That will be the order of presenting the image. I mean, turning a 2-dimensional, a 1-dimensional structure into 2-dimensional image. This looks kind of a simple structure, but it's not so simple. Yeah, this is, I'm just what I said. This is one of the reasons, and also there are 4 bases. So 3 positions and 4 values also indicate that 3x4 matrix will be the best basic image, 2-dimensional image, as we can see here. So this 3x4 or 12 positions matrix, 3x4 matrix, is a bit complicating in relation, for example, the neighborhoods of its elements. While the value structures of 5 elements that I presented to you has a linear neighborhood relationships. Only uracil and thymine have one neighbor, while all the other between have two neighbors, but it is all in essence linear structure, this spatial structure has three types of neighborhood relationships. As you can see, these corners here have two neighbors. It's this, two. So the neighbor is this, and neighbor is this. Neighbor is this, and neighbor is this. These middle ones have three neighbors each. And these central ones have four neighbors each. So this is much more complex structure than the value structure. And this is how it looks when we place in this spatial structure, the values. And we started with a dark gray, gray, light gray, white. And then we can go again, the dark gray, gray, light gray, white, until we exhaust all the possibilities in which each of the values will occupy each of the three positions. As you can see, here white is in the first position, here is in the second, here is in the third of the color. And the next will be a repetition of one of these, if we'll continue. So this also gives the reason why we have to have a 3x4 as a basic 2D image. So this we will call the D icon, and on the right end we see something that we can call the R icon. And again, if you compare the letter representation with the visual representation, you can clearly see the difference, visual difference. Now these are various kinds of, these are DNAs, D icons, and these are R icons. These are all corresponding each to other. But also this gives you the sense of how we can organize the larger structures based on these building blocks. These are like bricks. So we can build a larger structure with these icons. As, for example, the genetic code. And on the left, it's a DNA version. On the right, one of the DNA, because there are two for each of the helixes. And here, this is hand done. There are all three versions and one with the alphabet. DNA 1, DNA 2, and RNA in the alphabet representation and RNA in the visual representation. And here it is applied on this, I even forgot what it was a long time ago. It was called locus offen 2, but it is what is important, it has 2,300 bases. And I managed to arrange them in these kind of complex images, so you can see what we can get. Otherwise, it will be just letters, as we have seen, if we don't have this. These are the numbers, are the positions on the strand. Now, when we have spatial neighborhoods, we can now define two kinds of relationships when the values occupy positions, spatial positions. For example, if we have two boxes, like here, if we have in each box the same value, in this case white, we will call this connection. And for 4 DNA bases, there are 4 connections: GG, TT, AA, and CC. So this is a spatial neighborhood between identical values. Then we have something that we'll call junctions. Junction is a neighborhood between, a spatial neighborhood between two different values. In this case, we have thymine and guanine. They are spatial neighbors. Guanine and adenine are also spatial neighbors. And adenine and cytosine. And the difference between them is 25%. This is what we have on this main representation of the genetic code. Then we have cases when you have 50% difference between, you can have one position, one square in which there is white, and next to it could be gray. The difference between them is 50%. So that will be called the junction of the second order. And there are two with the DNA, between T and A and G and C. And there is a junction, only one, between white and dark gray, which is the 75% difference. And here you can see this is random, but to demonstrate how it will look on the SAM-D icon. For example, here white thymine is connected with the thymine once and twice. So we have 2 times TT connection. Or we have GG is here. It is connected once. We have 1 GG connection. And here we have, I think, CC as 1, and here is AA. On the other hand, we have junctions, like we have 4 times TG junctions. This is T, and this is G. 1, 2, on this side 3, and here 4. 1, 2, 3, and 4. And the others are, you can see. This shows you how we can, how to say, analyze these icons on the basis of their neighborhoods. There are some very specific, it's always like in all the sciences, especially like physics and math, physics and math is a language in a way. You reduce to the simplest cases in order to analyze the more complex later. And here we have a case of binary structures consisting of only of 2 values, thymine and adenine, in 3 distinct kind of a distribution. One is shown in these 2 cases. It's shown with the 2 cases. One case here is with clearly distinct fields of dark and white, which means gray and white. This one and this one. There is no white among this this gray, and there is no gray among this white. And it has a quantifying value. There are minimum junctions here. There are only 3 junctions. And there are 14 connections. This would be the state of high organization. In thermodynamics, this is a definition of the highly organized system. Like if you have this room divided in half, and then one will be very hot, another will be very cold. This would be kind of this room. And why it is highly organized? Because if I put a thermometer in each part of the room, I will know in which part of the room I am. If we remove the obstacle, and after a certain time there will be, entire room will become warm, we wouldn't know with the thermometer in which part of the room we are. That is called entropy. So this is opposite of entropy. This is the highest possible organized states with these 2 elements, an equal number of elements we have. Both, we have 6 T's and 6 A's. here is something that like a transition, like we are moving from the high organization to entropical case. This is some stage between in which we already see now C is not 14 but 9. Number of connections are going down and number of junctions are going up. So there are more connections between different values, less connections between more junctions, less. And then we have finally the state of entropy, in which there is no C is 0, no connections. And we have number of junctions is maximum. So this is how these spatial neighborhoods can be used to determine notions like an entropy. Now, some 2-3 years ago, let me find this. I've been in touch with the geneticists with this, and so one of the person that was I had a very intense communication for several years is Victor Norris. He's an American, but he is a theoretical geneticist at the University of Rouen in France. And he said, why don't you try this on this concrete biological existing E. coli strand and see what you can do, or what you will get. And the name of the strand is E. coli K12 MG1655. It has 4,639,675 positions. And one summer, 3 months, I spent extracting from these 4,600,000 positions only stretches of A and T's that are 12 and longer. 12 positions and longer. It can be done with a computer if we write a program in probably an hour. But I spent 3 months. And this is, you can see the setup. And here is, in fact, the stretch of 12 positions that I will put on the screen and identify 12 positions, as you can see here. So these are the positions. So this is, I was here, like say, this point when I took this picture, I was 1,702,990. That was where I was. And I would have, in fact, a piece of cardboard which has a cut that is exactly 12 positions long, and I will put it on the screen to check if this is a 12 positions. And that's how I will go through entire, and in this way, and this would be, for example, this is like, as you can see, this is 9,900, but it's like 991 because it starts with 1. It is TTA, TTA, AAA, and TTT. This is kind of a stretch I was looking for. And this is that position, and this is how it looks like what it looks on this government site. And this is how it looks like an image. And you can clearly see how different these are. These are now first recordings. This is how I recorded them as I was going through. I felt this is like a journey through DNA. And I felt that this is, I have a camera, that I'm going and filming through the DNA. That was my interpretation of it. In a way, and probably I will get later to you, what we are looking, one way to interpret, what are these images? These are images of the world recorded by DNA. If we understand that DNA is a living organism or proto-DNA, because what we see in DNA code today are echoes of the earliest living organisms that successfully survived, and we are the descendants. That must be in our genes. The first information about environment that were recorded by the first living organisms are in our genes. There is a complete continuity. So these binary cases might be the earliest images of the world that were recorded by these earliest living molecules. If some alien would, for example, take any of us outside of our planet, they will be able to reconstruct the environment in which we exist by our properties. Because they will find under what pressure, what light, what temperature we can survive. And they will tell, this is the environment in which this organism lives. Because these information are encoded in our body. The same thing is for the first living organism. First living organism had to survive to see where is hot and where is cold. In order to do that, it has to have a memory encoded about its environment, memory of hot and cold. Otherwise, it wouldn't be able to distinguish hot and cold if it doesn't have a memory of hot and cold. So what we are looking here is, in fact, the crude pictures of the environment recorded by the first earliest living forms. That's what I believe what we are looking at. And that's why these are binary structures. And I think A and T are the oldest one. So this is how it looked here. I mean, these are some pages. There, everything what I went through and managed to extract is in the book, entire research. So I managed to find something like 4,456 stretches, which means 4,456 images of this kind. Now, this is a second draft when I prepared them for publishing. It has to be done a little bit better. So each one has a position and the sequence, how it is today. And then you have above how it looks visually. Now, just compare this line with this line. Okay. Something is stopped. Let me see. Okay. I was particularly interested in the cases of equal numbers of A's and T's. And I found something like 700. Let me see. $774. So there are 774 cases in which we have equal number of A's and T's. And in some, we have these, as you can see, these are 4 cases of highest organization. There are 8 types of highest organization within this proposition, with this system, and I managed to extract them all. So there is only one I found of this, the simplest form, and that's on the position 1,064,135. And then this is the second type, which is like a C and type, which has 2, and this one has 3 as well in this kind. So there are 8 types of highly organized states.
Sorry, just to interrupt, but because it's too complex for now. So this is what you meant, binary structures in relation to the matrix of
Yes. Yeah. These are all 3 by 4. And for the 3 by 4, with 2 elements with equal numbers, we have 8 possibilities to organize them in a way that they are very highly organized. And these are here, as you can see on the left, like this line. This is one type. And it can be in this way, this way. These are variations of the same type. And this is another type with the F kind of a form, as you can see on the number 7. And this is this kind of L type, which is number 6. So, and they all have the same number of elements, 6 by 6, equal number of elements. And this is like a P. So there are 8. There are no more, no less, only 8. And there are 2 kinds of highly disorganized or entropical, which I have shown you 2 chessboards. However, I didn't find any chessboard, any entropical sequence. Only the closest one was this one. However, on this, I even cannot pronounce aspergillus stretch, or I mean, not stretch, it is mushroom. There are very strange, and this is very short one, 92,120. There are not only the chessboards, many cases of chessboards, or means high entropy, between C and T, but also we have these blank, completely gray states, or here completely white, which I don't know what does it mean, but that's all like this. Look at how the line looks like. It is A completely until here, and here starts T, which is this one here, white, and then it enters, there are a few more T's. So that is one thing, how to distinguish highly organized or low organized states. Another what I found interesting by doing this is how these states relates to each other, how these images relate to each other. These are neighboring images. As you can see, this is the position, this is the first stretch that I found, position 96, very beginning. And as you can see, they are all done in a way that first image is we start with T, which is here white, and then we go AA, which is gray, and then we go ATT, which is gray, white, until we get to this position 7 here. That is the first image. Now, and that is like, imagine like a frame of 12 positions. Now we move the frame just one step. And we get this position, this picture. And this picture is not very, it looks not exactly continuum from the previous one. And then we have this one, also next step, and this one next step. You can see some structural similarities, but it's also very complicated. It's not clear on the first sight. It was not clear to me. And then I realized that if we compare these 2 images, we can notice that this vertical line, dark, white, and this vertical line, dark, white, move to the front on the next picture. At the same time, the first line, 3/4, move to the back, on the top. The only thing that left is this, and the only thing that enter is this new T here. That's why we have, instead of 2, we have 3 T's here, 3 white. So in effect, again here, look, only these 2 lines, 2 verticals, this one and this one. It's like inverted C, and it goes to the front completely. in its entirety. And this first line, the dark goes out, these three positions goes, three positions here goes here, and the new T enters. Now, I was thinking, what if this is all, if we think of each image is an instance of a present, now, then these are three components of now. One component we can call the past. This is the present, and this is the future. In a way, if we look here, I will show you in the next thing, on these three images. In this, if this is present, and this is a future that is coming, and this is the past that is going, then in the next instance, what was present becomes past. What was future becomes present. And what was past, at least part of the past, becomes the future, with some new details. Doesn't make sense, but I think it will make sense in many ways. So the past is not completely gone. Past comes back, but not completely as it was. It has some new, it has an iPhone, for example. So that is the new element that entered. This is like a schematic representation of this. In the first instance, this is past altogether. This is present and this is future. Now, present and future moves one step to the left, and this is simply by moving one step of frame, nothing else. That's what is really amazing. And another, so now one way of using this grayscale, and I think that we have a grayscale as our kind of something that is very familiar to us. It is because it is a genetic code. It is encoded in our genes. That's why we have this as something very familiar. What we are looking at is, in fact, the what is the code? The code is not A, C, G, U, and so forth. The code is black, dark gray, gray, light gray, and white. And this formula can be applied on various other phenomena. Like, for example, this 5-element structure can enable us to structure the word quantifiably, in a sense that we can say gray is, I think this is a bigger picture, doesn't matter. The gray is a referential one, it's 1. Then light gray is finite small, and white is infinite small. On the other hand, we have dark gray is finite large, and black is infinite large. So that is one way, and you can apply to different properties, like time, or you can apply to distance. These quantifying elements. So entire universe we can structure in these 5 elements. Or the relationship between observer and observer. If we say that, and this coincides, as you can see, these subsets of this grayscale, observer is from the dark gray to white, and observed is from the light gray to the black. You can say that in this case, there are parts that both observed and observer share. In a way, we can say that dark gray is known universe. The gray is interface. It's, for example, our eyes. That's what is gray. Exchange between outside and inside. And white is, light gray is our center for processing the visual information. They all belong to the observed at the same time. They are part of the world, but remains part of the observer that will never be part of the observed, which is usually I. So this indicates that I is not part of the observed. As well, on the other hand, it says here that there is a part of the observer that will never observe, will be observed, that will never be known. So these are completely separate, and they come out of this uracil and thymine. And because why we can structure the world this way, or relation between, is because it is already structured in DNA. And another is also life and death. Life and death are the binary situation, life, death. But in this might be that this is not exactly such a binary situation. That might be the stages between that are part of the death that are still alive, or part of the life that are already dead. So this is how you, for example, I can give you so if you have in the mathematics x equal y through z, with this simple mathematical formula, you can define velocity, acceleration, you can define the relationship between resistivity, current, and voltage. You can define between frequency, speed of light, and wavelength. The same formula, if you give a different meaning to the symbols, you can relate to different kind of realities. In this case, this 5-element grayscale can be used to address different aspects of reality. And I believe the way we can do it is because they come from DNA. They're encoded. So in my way, this I just want to show you this, and I will tell you at the end, because this is the end now. So these are some of the images of this exhibition that Amal mentioned that was it was 15 years ago. Genesis, this is from Berkeley, how this material was. This was in Berkeley. And this is in Henry Art Gallery. There was no effect of this. This was just shown, nobody really paid attention to it. And there was also written, there are some coverage of this in a couple of articles. And this was the most recently again shown because of the book that was written, that I decided to write. Because there was a negotiations with some, I mean not negotiations, exchange with some geneticists to do a program that will, how to say, implement this method, so you can have any DNA stretch and just simply you type it and you get the image out of it, which is absolutely possible to do. And one of the person that was involved was David Ussery from he's head of the laboratory in Oak Ridge National Laboratory. As you can see, he likes the book, but he's head of the comparative genomics group at Oak Ridge National Laboratory. He keeps the book because he likes it, but nothing happened with it because he's a biologist. He doesn't know what to do with it. Because this is not, I mean, this can have an implementation in biology, but this is not about biology. In a way, this is not about what DNA does. That's what biology is. It is what might it mean, what might be the meaning of DNA. That is here the emphasis in this work. And I also figure out that this, what we are looking here, is the code. This is the DNA code, DNA RNA code. This is not the DNA RNA code. This is completely arbitrary set of 5 alphabet letters. Because if really, as I said, the first DNA, proto-RNA, it was most likely proto-RNA organisms, the important thing for their survival was hot and cold. To find out where is hot and where is cold. And hot and cold are black and white, dark and light. That is the hot. So this is the code. And also in another way, this is not invention. This is a discovery. It is closer to periodic system of elements, kind of a discovery, than it is an invention. When I'm saying that this is discovery, this is the code. That's what I mean. And this is the approach that treats DNA as an observer. And what is encoded on DNA are information about the environment that was necessary for its survival. I mean, the proto-RNA that was then transferred, I'm just simplifying this. So this is what I had to say tonight. I see nobody left.